It's my pleasure to introduce our speaker today, Daniel O'Gorman. Dan is one of our CCOVI professional affiliates, but his main job, where he resides, is actually in British Columbia. He's a research biologist at the Agriculture and Agri-Food Canada Pacific Agri-Food Research Centre in Summerlin, British Columbia. So we're very pleased to have Dan come out and spend some time with us over the next couple of days and share research talk today. So Dan holds a Bachelor of Science in Biochemistry, yay biochemistry, I'm a biochemist too, Dan, from the University of Victoria, and a Master's of Science in Plant Pathology from the University of Guelph. His research focuses on the development of molecular tools to study both beneficial and pathogenic microorganisms, and that's fungi and bacteria for those that may not be familiar with those microorganisms, to help understand and manage their impact on plant health of wine, grape, and tree fruit crops, and also to identify the relationship between agriculture and microbial biodiversity to maintain healthy agro-ecosystems. So with that, Dan, welcome today, and we look forward to hearing from you.
Thank you. Good afternoon. I want to thank you for inviting me out here, and I think the last time I was here a couple of years ago, I brought sunshine with me, and I was told this is more BC weather that I brought with me this time, so it's getting better, I understand. So my talk this afternoon is going to be on grapevine trunk diseases, young vine decline in particular, and the development of a DNA macroarray as a diagnostic tool. For those of you not familiar with macroarrays, they've been around for quite a while, the last 20 or so years, and have been used quite a bit in the medical field, but also have been used in agriculture. And the macroarray is the poor boy of DNA arrays. And there's a couple advantages to going this route as opposed to the more upscale DNA chips or gene chips, microarrays. The difference with the macroarray is it's cheaper for one thing. It can be produced on in a lab without very sophisticated equipment. So just a basic plant pathology lab or microbiology lab could put this together, and the array would be about the size of a business card. And on that business card, which is a nylon membrane, you can have hundreds of different probes, DNA probes, which will bind specifically to different organisms. So now, if that compared to a microarray, which would be the size of your pinky fingernail or smaller, would hold thousands of probes that could detect different targets. Much more expensive, but the technology is the same and it can be transferable either way. I hope through my talk you'll understand why we're looking at producing and designing this DNA array for these organisms, and I hope that becomes clear. There's a couple of things before I get started. I'm going to use the word DNA array or macroarray and membrane interchangeably, simply because on our macroarray we use a nylon membrane as a support for the array of probes that we have on there, so I don't want to confuse anybody with that. Secondly, I'm going to use the royal we a lot, and that usually means that the postdoc in our lab, Jose Urbez Torres, Paula Haig, and Megan Sheepwash in our lab as well, summer student, have all worked hard in this and taken the lead on the work I'm going to show here. So, before I get, how do I, there we go. Thanks. I'm just going to give an outline of where the talk is going, just so you can follow along. I'm going to give a snapshot of the Okanagan Valley in British Columbia and the wine regions or areas that the production occurs in BC. I'm going to follow that up by talking about grapevine trunk disease and young vine decline, and just the impact that it has on grape growing in British Columbia. Then we'll get around to the DNA array system. I'm going to explain that a little bit, how it works, and then the development, what we've done, and what we've developed so far, and looking at diagnostic results and further testing of the array. And then we're going to look at the potential of this application and the ongoing and future work on the DNA array. So here, just a picture of Park. This is Pacific Agricultural Research Center in Summerland. And we have about 320 hectares of land there. 90 to 100 is irrigated and in production of tree fruit and grapes. And I think roughly 100 to 150 people are employed there, depending when the summer students come in. Big influx in the summertime. Another shot of the facility there from above, looking over the cherry block and down on the lake across at Naramata, the Naramata bench. And the reason I wanted to show this picture in particular, because you can see those silt cliffs, and I've been told this is the remnants or lake bottom left behind after the last ice age. So, 10,000 years ago when the ice receded, the lake was a lot higher at that point, and what you're seeing there is lake bottom. As you move up the valley hill, up into the hills, that can often change into sand and then gravel as well. And of course that's a generalization, because I've been to some blocks where a 5-acre block can have 5 different types of soil types in it. So, but in a general way, that's what it looks like, and quite often that's what you're seeing up and down the valley. Oops. So, grape growing in the Okanagan, some of the desirable conditions that we have grapes or ideal growing degree days with lots of sunshine, low rainfall and low humidity. So we actually get a lot fewer rocks, so the diseases, bunch rot and sour rot, that kind of thing that you'll have to deal with out here. So, that's a real plus. Some of the extremes, we have a fairly short, compact growing season, probably very similar to what you guys are experiencing here. We also have variable weather, especially noticeable in the wintertime with these cold Arctic outflows sweeping down into the northern Alberta and Yukon, and temperatures dropping down to minus mid-20s. But we also have warm ocean weather that can come in from the Gulf of Alaska. So the grapes never know whether they're warming up or cooling down. So becoming acclimated to the temperature is a bit of a problem, especially when you get those cold snaps. Just the geography of where we're situated. Smack in the middle of the province, right along the border with the U.S., about a 5-hour drive from Vancouver. And we're right in the rain shadow of the coastal mountains, and yet these valleys, the Okanagan Valley has been carved out by the glaciers, quite narrow topography, and yet we're far enough away from the Rockies that we don't get more precipitation as the clouds bump up against those mountains and drop more precipitation. So we're quite dry. We have good continental heat that develops during the summertime. But both summer and winter, the conditions are moderated somewhat by the lake effect. Okanagan Lake is big enough that it really has an effect on our temperatures. And as I said, that warm Pacific air can sweep in, as well as cold Arctic outflows. So quite variable weather can be experienced there. I've got the Okanagan is the asterisk there, or the big square. From the South Okanagan to the North Okanagan. Above that is the Shuswap area, which is just shy of the 51st parallel. And so I think the most northerly vineyard in British Columbia is up there, as that's what they have on their website, and it's probably true. Possibly one of the more northerly vineyards that you're going to find in North America, but I haven't checked that out. And the varieties are growing up there, obviously something that's going to ripen early, Rieslings, Gewurz, I think Ortega and Foch are also grown there. Similar to what they're seeing on the coast, because of just the amount of heat that they get down there, they'll need varieties that will ripen earlier. And also in the out east, right out there, you'll also, in the Kootenays, there are a little bit of grape production starting to pop up there as well. And this just shows a map, although that may be a little hard to see. We have the Okanagan Valley starting at the U.S. border of the Soyuz, all the way up just past Vernon here, about 150, 200 kilometers north of the U.S. border. The Similkameen region also has grapes growing in it. The Kootenays here, again, some more grapes being grown up in the Shuswap area. So most of the production here, I think, as I'll show you in a second, is going to occur in the lower part of the map in the southern Okanagan, so from Summerland south. But just before we get there, this is another bird's eye view of Okanagan Lake with Summerland right down on this little bump in the lake, and as you drive up around the corner in the top right-hand corner, you'll see about an hour, 45-minute drive from Summerland. I borrowed this slide from the BC Wine Institute just to show where the grapes are produced. Unfortunately, it's a little blurry. Didn't copy quite as nicely as I thought. But you'll see that most of the production, again, in the South Okanagan, as I mentioned, with the bulk of that in the Oliver area, Soyuz, Penticton, OK Falls. Summerland has a small little wedge up there. The remaining little slices are going to be Kelowna, the Shuswap area, along the coast on the Gulf Islands, Vancouver Island, and out in the Kootenays. And what you're seeing as far as production, most of the reds are going to be down in the south, obviously, where most of the heat occurs. But for the white variety, Pinot Gris, Chardonnay, Gewurz, Rieslings are popular varieties that are grown there. And for the reds, Merlot, Pinot Noir, Cab Sauv, Syrah are among the most popular that are grown. Just switching to trunk diseases. These are diseases caused by fungi, Ascomycetes and Basidiomycetes, so two very large groups of organisms that are involved in this. They infect the vines through pruning wounds, so, well, primarily pruning wounds, but any wounds are going to be openings for these organisms to get into. So any mechanical wounding or even winter damage. So if you have any splits or cracks in the trunk from winter damage, the spores are going to get in there, be able to get in and penetrate and colonize the wood that way as well. So they're said to be endophytes or facultative parasites. So part of their life cycle will be growing in the vines without causing any kind of disease symptoms. Now, it's really hard to find out where this comes from in the literature because I haven't found it yet, but this is a fairly well-accepted notion that these things exist in the vines without causing any problems until something triggers, some sort of stress response triggers disease to occur. So I think that needs to be investigated along the way to nail that down and see where that notion came from. Once they gain entry into the wood, they'll colonize the vascular tissue, either causing cankers, which canker diseases are Botrytis dieback and type of dieback, or you'll also see this black spotting or streaking of the vascular to the xylem tissue, and these are the ESCA and young vine decline type symptoms that you'll see. And once they're infected, depending on what species it is that's infecting the vines, you're either going to see a rapid death very quickly over a several-week period. The vines would go from being very healthy to dead, and usually during the summer heat. Or you can have a slow decline over several years, 3 to 4 years. You'll just notice that there's delayed growth in the spring, and they have a hard time catching up. The next year, it's just a little bit worse. 3 or 4 years later, you'll have an eventual plant death. So they obviously cause a significant impact, economic impact on the grape industry through loss of yield, and then plant death and replanting of the vines that have died. Young vine decline, so just looking at vineyards that are less than 5 or 6 years old, and it's caused by Petri disease and Blackfoot disease pathogens. And most of these, you're going to see small plants that are just struggling to survive. Sometimes the leaves are necrotic or chlorotic looking, just the vines, there's the delayed bud growth or growth in comparison with some of its neighboring plants. Some of the vines will also produce foliar symptoms where the leaves go red, which is an indication of vascular dysfunction. And you can see the cross-section of the vine in the bottom. This is very typical of what you're seeing when you cut into the vines that are affected with young vine decline, either Blackfoot or the Petri disease pathogens. Blackfoot will also cause black sunken necrosis in the roots as well as this dark streaking. But other things can also cause these kind of symptoms. As you can see, there's a picture of a J root here, so improper site selection or preparation. You know, if you're planting shallow holes on hard pan or something, the roots have nowhere to go, very shallow, they dry out, and also causing, obviously, vascular stress can cause very similar symptoms, so it's not always clear-cut what you're dealing with. So for mature vineyards, you're going to find the ESCA diseases, Eutypa, Botrispharia, and Phomopsis dieback. And the symptoms there, both foliar symptoms with the tiger stripe for ESCA, as well as the measles that are seen on grapes. Primarily table grapes will show these symptoms. But you can also see these chlorotic, shaggy, small, misshapen leaves that go along as a symptom of Eutypa, as seen in the lower center picture here. Very characteristic of what you're going to see for Eutypa. And again, you can either have the dark streaking or the canker production for these diseases. So to identify what was the impact of trunk diseases in British Columbia, we did a survey where 400 samples were collected through vineyards, through the Okanagan, Similkameen, up to the Shuswap area, down on the coast, Vancouver Island, Gulf Islands. And these samples were brought back, and we looked at the vascular tissue to see what samples, excuse me, what the samples looked like. And here's just an overview of everything here, and we can see that there's three main problems that we have: the dark streaking of the wood, the wedge-shaped cankers, and also a mixed infection where you have canker and the dark streaking occurring in the same sample. So these three were the most predominant of all, about 30%, and no significant difference between those, with these internal circular cankers showing up, but at a much less rate of about 12% of the vines showing these symptoms. If we look at just the young vines, we're going to see that the whole scenario changes up a little bit, with 67% of all the young vines, less than 6 years old, of the samples collected were showing this dark streaking of the wood and plugging of the vascular tissue. And these were occurring primarily in the rootstocks, with 87% of all the samples showing the rootstocks with these dark streaking symptoms, as compared to the wedge-shaped cankers, which only 19%, and usually occurring up in the sign. There was none of the circular cankers shown in any of the young vines, and only 13% of them had mixed infections. If we look at the more mature vines, we can see again it changes up. The dark streaking of the wood decreases dramatically down to 14% from 67%. We have an increase in the wedge-shaped cankers up to 33%. Circular cankers, 18. Those are the internal ones. 33% of these vines had mixed infections. And most of these, especially the major ones, we're seeing in the cyanid, so above the graft gene. We also, part of the survey also looked at just field symptoms. So what do the vines look like? What were they expressing in the field as far as canker development or foliar symptoms? And so Jose also surveyed same areas throughout the Okanagan, Similkameen, Lower Mainland, Vancouver Island for field symptoms of grapevine trunk diseases. And how we did this, we flagged an area of 10 rows by about 50 vines and walked them and identified each vine and indicated which vines were expressing symptoms, and And these were either necrotic or chlorotic looking leaves being expressed, stunted growth, actually completely collapsed vines, or the shaggy leaves with typa, cankers, etc. And once they're flagged, it was easy for us to go back later in the season to see if this has increased at all, if the number of plants showing symptoms have increased, or even following years we're able to return here and see what these individual blocks look like. And so that'll give us a good idea whether the situation is really stable or whether or not the diseases are spreading. And if we look at these, well, in this, we only have looking at the young vineyards within that survey, 44 young vineyards. We see that 3.8% of all the vines surveyed were expressing some sort of grapevine trunk disease symptoms. And over half of those were young vine decline, the other half being made up of canker diseases and ESCA type symptoms. The same 44 young vineyards, 44 vineyards, 7 had no symptoms at all, where 34 of the 44 showed symptoms in vines, 0 to 5% of the vines. 2 vineyards showed between 5 and 10% symptomatic vines, and 1 vineyard had greater than 10. And in that particular vineyard, it was almost 20% of the vines in the vineyard showed symptoms of young vine decline. If we look at, again, the same 44 young vineyards, but at ESCA symptoms, we see now that almost 36% of the 44 vineyards had no symptoms of ESCA, where 2 vineyards had between 0 and 5%, 1 vineyard had 5 to 10% symptomatic vines, and 5 vineyards had over 10%, and these ranged from 13 to 18% of symptomatic vines showing ESCA symptoms. So once the survey was complete and we saw what the symptoms looked like, we had an idea what was occurring out in the vineyard. We wanted to see what species were identified or connected to the different symptoms that we are seeing. And in order to do this, we take the samples, cut out small pieces of tissue, infected or diseased tissue, plate them on growth media, grow them up in the lab through several culturing steps. We can come up with some pure cultures of the different fungi, and then we can identify those based on colony morphology as well as microscopic examination of the spore characteristics. Now, if you're really good, and Jose's pretty good at this, you can get down to the species level sometimes. But most times we have to use DNA sequencing to really nail it down, confirmation of what we're looking at, what species we actually have. So we do use DNA sequencing of the pure cultures in the collection to get confirmation of what species we're dealing with. Just a phylogenetic tree of the diseases or the pathogens that we found in the survey. And what this shows us is the genetic relationship between all the organisms that we've identified, not only between the different groups of organisms, but within the same group. So for instance, the ESCA pathogens, we have a group up here, I have to read it on this slide, the Phaemonniella group, so causing ESCA symptoms is up there, but we also have ESCA pathogens down here. So very different organisms genetically, morphologically, but they're causing the same disease. So it's important to know what you have and what species is causing it. They can react quite differently. Some are very pathogenic, some are less pathogenic. So the reason to identify what pathogens are involved is important for this reason. And what we found from the survey, 28 different pathogens exist in BC and have been confirmed there. And we have the young vine decline and ESCA pathogens, Blackfoot disease pathogens, Botrosphaeria dieback, Typa dieback, and Phomopsis. So 28 in all known pathogens that we have been confirmed in British Columbia. But we also have a second list of organisms that are potential pathogens. So they're all well-known pathogens on other hosts, tree fruit as well as berry crops. and they all cause canker or dieback of these. So they're of interest because the frequency that they turned up in the survey was relatively high, so they really deserve a second look to see whether they're truly pathogenic on grape or whether they've just come in as kind of a secondary infection, opportunistic, once a plant has been weakened. And here's where we connect the actual symptoms with the species. So out of the dark streaking symptoms of the vascular wood that we see, Phaemonniella cylindercarbon, Phaeacromonium species are kind of what we would expect to see from this symptom, but we're also seeing Botryopsferia coming in here, and the Botryopsferia is one of the canker pathogens. So obviously it's a mixed infection, and it's interesting to see that we're isolating it from this type of sample. The bottom part of the slide shows the wedge-type cankers, and with Cytospora showing up as the #1 pathogen here being isolated from these samples. The interesting part there is that Cytospora, although it's a canker pathogen, not on grape. It's going to be found on tree fruit, but it's being isolated out of these samples at 27% of the time. So obviously one of the ones that we want to look at and do some pathogenicity testing and find out, is it truly pathogenic on grape, or is it just coming in as a secondary colonizer? Below that, the Diatripaceae and Botryopsferiaceae are going to be your Bach canker and Eutypa. And again, they would be what we'd expect to see with these type of symptoms. The circular necrosis, or central canker, Diatripaceae, the Eutypa species, if you look in the literature, you're not going to see it associated with this type of symptom. So this is something new that hasn't been described before, and yet it seems to be quite common in BC. Cytospora present again in this as well, a non-grape pathogen. Phaemonniella and Botryopsferia, Phaeacromonium species, all present when you see these type of symptoms. So describe, it kind of brings home the message that this is definitely a disease complex, multiple pathogens involved, and can be associated with different disease symptoms. And to be able to tweak these apart, you can't go into the field, find one symptom and say, one pathogen. It's a little trickier than that, and you have up to 80 different pathogens worldwide that can cause these. So again, this is part of the reason we're looking at a macroarray to help us with the diagnosis, because one test can identify any one or all of the pathogens that may be present in the sample. And here we are, the development of the macroarray. So I got into a little bit of this before the talk. I'll just repeat it again. The macroarray differs from microarray simply by size. We're working on a nylon membrane, and it's a hybridization reaction where we apply sample to the nylon membrane, and DNA from the sample will bind to the probes that have been put on to the membrane. And on our membrane, we can have hundreds of probes arranged in a grid pattern or an array of probes on the membrane, and each one of those probes will be specific for a different species of pathogen. And that differs again from the gene chip, which can have thousands, but that's just for comparison. What happens when we have the binding of the DNA, sample DNA with the probe, is that it gives off a chemiluminescent response or reaction, and that can be captured on X-ray film or a digital camera, and then we can identify what probes are reacting and then what species they relate to. And again, the power of the DNA array comes with the ability to identify diseases that may exist in a disease complex. So if you have more than one disease causing symptoms, you can tweak them out to identify all of them that are responsible. And again, with large numbers of pathogens, we can identify one or all of them with just one single test. So it takes the guesswork out of it. What test are you going to use on what sample to identify what's going on? Now this is an example of another macroarray. It was produced by an Ag Canada researcher in Ottawa, Andre Levesque, and it's been licensed to a company in the Netherlands in support of greenhouse nursery industry, and as well as the turfgrass industry in Europe. And they're calling it a DNA multiscan. It's basically what we're doing with the nylon membrane and probes on it about the size of a business card again. I just threw this in as an example of what it's done and what kind of industry it's supporting and the utility of it. So what we're doing is looking at young vine decline and young vine decline pathogens. All the ones that have been identified in British Columbia, as well as all the pathogens that have been identified around the world in other great growing regions. But we also wanted to include closely related species, so related species that aren't necessarily pathogenic on grape, but within the same genes, just so that we can rule out, well, just ensure ourselves that we're capturing everything. So they may not be known pathogens yet, but if we see them occurring in grape, we'll be able to pick them up with the membrane. It also allows us to use this membrane for taxonomic purposes as a tool in scientific research. So there's a number of reasons to go that route. So these numbers here for the Phaemonniella, the Phaeacremonium, the Cylindercarpin, the Camplocarpin, the Aleonectria, those are all the Blackfoot and Esca type pathogens that are involved in young vine decline, and also the related species. So in total we have 86 young vine decline specific probes on the membrane, as well as some control probes that are put on just to as internal control, so we can tell what's going on with the hybridization reaction. So to do this, we had to put together a fairly large collection of fungi, and we have probably assembled one of the largest and complete grapevine trunk diseases pathogens, fungi collection in the world, with all the X-type cultures. And now the significance of the X-type culture is those are the isolates that were used by taxonomists when they described individual species. So as far as referent cultures go, they're about as good as it gets. And we have X-type cultures for most, but not all. And where we couldn't obtain X-type cultures, we were actually able to obtain either cultures or DNA from other research labs around the world, and that we're now using for reference cultures. But as I said, most of these are X-type cultures, and it's going to be a great resource for us for future research. The last slide, these are the Phaemonniella species involved in Esca, both pathogenic and non-pathogenic on grape. Phaeacremonium species, again, both pathogens and non-pathogens, 35 species in all, with 58 isolates. And the Camplocarpin, Cylindercarpin, Aleonectria, these are your Blackfoot pathogens, and again, pathogenic and non-pathogenic isolates, 15 species, 37 isolates. And so these come from all around the world. We've covered almost all the grape growing regions in the world. So we have quite a complete collection of reference cultures from worldwide collections and sources. And so this will put us in a pretty good position to produce the membrane that we're, or X-ray now, the membrane that we're producing now, as well as future membranes on the bot canker diseases. So what we did with the cultures, extracting the DNA and sequencing them, and we used the beta tubulin gene down near the 5' end of the gene, and it straddled both exons and introns for both conserved regions and variable regions of the gene. And the conserved regions allow us to kind of anchor the sequences together, and the variable regions are going to be where we're looking for differences that may exist between the different species. And those variable regions are what we're going to be using to design the different probes for the individual species. So once the sequencing is all done, we do DNA alignments and we create large text files, and you can see that we have in this large regions here where all the sequences for all the different species are the same, but over here you start to see gaps in the sequence alignments, and those have been highlighted in the boxes, and those individual boxes that highlight the differences actually correspond, if you could read the names of the species over here, they would actually correspond to individual species. So the differences you're seeing are mutations, insertions, and deletions that may occur between different species and allow us to identify unique little sequence tags for each individual species. And from these, that's what we create our probes from. Oops. And as I said, yes, they do correspond to individual species that we then design probes for each of these. So each species will design one or several probes, which are little DNA, pieces of DNA that match the exact sequence within those boxes. And then each probe is specific for each or an individual pathogens. We send these sequences off. They're synthesized for us by a company, and they also put a little linker arm onto the sequence, and the linker arm here just allows us to attach the probe to the membrane, and it also holds it up off the membrane during the hybridization. It just allows for a better reaction to occur. So once we get the probes back from the company, we hydrate them in a buffer and a little bit of dye just so we can see what we're doing. The probes then go into a 384 well plate, and we use a pin apparatus. It looks very much like a hairbrush, little metal pins that we dip into the plate, and then from there we transfer or print that onto the membrane. So we're transferring about 0.2 to 0.5 of a microliter on the end of each little pin head onto the membrane. As you can see on the membrane, the little blue dots each represent one probe. Each probe is unique for a different species. And this is just an example of a grid or a membrane, the way the probes are laid out to identify each species. And when you have a positive reaction, you're going to have gray dots that are produced when you're exposing these membranes on an X-ray film or to a digital camera. And the way that occurs is that we've taken sample DNA and amplified it with PCR and simultaneously labeled it with a DIG DUT molecule. So that gets incorporated in about once in every 10 T's that should be there. You're going to get one of these DIG DUTPs instead of a T. And that takes place in the amplification process. DNA is then applied to the membrane for hybridization. And that label allows a chemiluminescent reaction to take place. If there's no reaction, there's no probe binding any of the probes, they remain blank. There's no chemiluminescence occurring, so you're not going to see anything on the membrane. Just to describe that whole reaction, the chemiluminescence a little bit, you have a DIG molecule attached. This is your DIG molecule here, and it's attached to your alternate DNA-based DUTP, which is incorporated into your PCR amplification product. During the hybridization, once the probe has bound to the sample DNA, we added an antibody that's labeled with an enzyme. So the antibody is specific for the DIG molecule, and it'll float around in the solution until it finds that DIG molecule, and then we'll apply a substrate for the enzyme that's linked to the antibody, and that enzyme then, in the presence of the substrate, will emit these little photons of light energy, and that's what we capture on the X-ray film or a digital camera. So your PCR product floats around until it hybridizes with a probe. Then we have the antibody. It looks for the DIG molecule that's been labeled, and it gives off little light energy that's captured on your X-ray film. And this is an example of some results. So this is your X-ray film, and as you can see, when you overlay the grid pattern on that, you can differentiate different species based on where what probes are lighting up. And in this case, we're differentiating two closely related species of Phaeachromonium, Iranum and Occidental. Slightly different template, so there's different prototype as we went along. Jose's looking at testing different control probes of the membrane. So we have control probes along the outside, here and here, with the inside of the membrane or array containing the species-specific probes. And this is just looking at differentiating Phaeachromonium species with closely related species, again, both pathogenic and non-pathogenic. And we're seeing that we have nice, clean probes lighting up, and in this case, these are the probes that were specific for the species. And in the other ones, you see that different probes are lighting up in different areas, also specific for those species. And this is a new species that we found during the survey. So Jose's working on describing this right now and getting a publication out. But it also nice clean separation and differentiation of the species. So the blackfoot pathogens, same sort of thing. Control probes along the outside that were being tested. Internally to those, you can see probes for the different species of Ilionectria here for differentiation, as well as cylindercarpin. Also, nice clean separation. There's no cross reactions occurring, so we're quite confident that these can separate the different species out quite cleanly. Phaemonella chlamydospora, again, here we have both the X-type culture looking at what probes are lighting up for that, as well as the park isolates, so ones that we've identified and isolated in British Columbia. And you can see slight differences. With the type culture, we have these probes that are lighting up, and then subsets of that same grouping for individual BC isolates. So as long as you're getting those probes, any one of those probes lighting up is going to tell you that you have Phaemonella species here. So it's not a matter of all or nothing, but because there is going to be some variation, some of these species, the type cultures may be coming from South Africa, so we have that regional difference that could exist in the genetic makeup of those organisms. So that's why we've created a bit of a redundant set of probes so that we can ensure ourselves that we are capturing everything. And then we just compare that to some non-pathogenic isolates, and again, nice clean separation and differentiation. We wanted to see how sensitive the array was as well, and to do this we set up a 10-fold dilution series of DNA for Phaemonella, Phaeacromonium, and Ilionectria species down to 10 to the minus 8, and then we amplified those samples and labeled them, and then hybridized those amplified samples to the membrane. And we can see that we have down to 10 to the minus 6 nanograms of DNA giving off a signal. So this is quite sensitive. That's down to femtogram amounts of DNA, or parts per quadrillion sensitivity. So, pretty darn sensitive. We also wanted to look at wood tissue and soil tissue that have been inoculated with these pathogens, and whether we could use the membrane and direct processing of these different tissues, so we don't have to culture them out and spend 5 weeks growing up in culture before we can extract DNA. So what we did is we inoculated wood with these organisms as well as soil, incubated the wood for 31 days before we extracted the DNA, and the soil was incubated for 10 days before we extracted the DNA, amplified it, and then hybridized it with the membrane. And with the wood, you can see the different samples here. This is just our agar plug, so that's our control. And all the rest here have been inoculated with one of the different species that I just named. What we would do is just carve out, just on the margin of this necrosis, carve out a little piece of wood, probably about half a gram, 200 to 0.5 of gram of this wood tissue, drop it in a tube, extract the DNA, and then amplify that, apply it to the membrane. And these are the results. So all positive results for the different organisms that we inoculated into these canes. And we can see both here the Phaemonella chlamydospora, which we have the type culture as far, and also the park isolates. We have Phaeacromonium species, again positive results for that, and the Ilionectria, Leodendri, again positive, and being able to distinguish each pathogen from the other within the canes. And then on the end is just our agar plug. And the only thing we have showing up there are the controls which run along the top and the side. So again, no cross-reaction between the species, and it allows us to extract the DNA directly from woody tissue. So we bypass the whole culturing step and go directly to identification of what species may be within any given piece of woody tissue. So I'm not going to show you the soil samples, but again, that was similar results for that, all positive and very clean. And we're going to sum it up a little bit, just to say that 59 different species are being able to be identified with the membrane. These include Phaemonniella, Phaeacremonium, Camptocarpon, Cylindrocarpon, Amelia necra, all the Esca and the Blackfoot pathogens that are involved in young vine decline. There's no cross-reaction between any of the probes that we designed and tested, nice and clean, and good separation between the species. And we have a lower detection limit, down to 10 to the minus 6 nanograms or femtogram amounts of DNA. And we're able to detect pathogens directly from wood and soil. And also, that's a big step. It takes up the whole culturing step, which can take up to 5 weeks for some of these species. And so what are some of the potential applications? Scientific diagnostic management as a management tool, all of the above, I think. As I said, the strength of this is that we can detect one or many species within a single sample. And it takes the guesswork out of it. If we're looking at doing taxonomic work, we can use it for that because we have included not only the pathogenic but closely related species from around the world. Epidemiology studies looking at spore loads in the vineyard. Diagnostics as well. Plant certification is another possibility for this type of tool. Another thing we wanted to look at potentially is looking at disease thresholds. What kind of spore load or disease or pathogen biomass does it take to create disease symptoms? At this point, we could use the membrane to identify what species may be involved in any disease symptoms, and then couple that with real-time PCR to identify what the actual quantification or spore load that maybe exists within each sample that is required to cause disease symptoms. Just back up a bit. The other way to do that is if we can figure out a way to get the membrane to be semi-quantitative, so low, medium, high levels of infection. So it won't be as nearly as specific as real-time PCR, but it may be enough to develop some threshold limits for some of these diseases. So that's something that we're going to look at as well. Our next step is that we want to produce another membrane or disease macroarray for the diseases causing cankers on grapevine. These are Eutypa and Botrytis species. But as well as those, we also have about another 30 species that include both Phomopsis and Cadaphra. So these species all produce cankers, so we'll have about 70 different species represented on this next membrane. And that will be coming shortly. As I said, we also want to look at whether or not we can get these membranes to be semi-quantitative so that we can look at disease thresholds and that type of thing. Again, potential applications. As a scientific tool, we'll be able to use this as taxonomic purposes for epidemiology studies, disease threshold studies. Also as a diagnostic tool, it can be transferred to public or private diagnostic labs. And as a management tool in nursery propagation and new plantings, looking at soil prior to planting. I don't know what the situation is here, but most of the vineyards going into BC are going into old apple plantings or old cherry plantings. And in the case of the Cylindrocarpon or the Blackfoot diseases, many of the same pathogens that affect grapes are also present in apple and cherry. So it'd be good to know what the disease threshold or the disease load is in these soils before you're putting in new vines. It can also be used in mature vineyards as a monitoring tool, just monitoring spore loads for disease management purposes. And I think that's all we have. So I'm going to stop it there. I'd just like to thank again the research team of Jose, Paula, and Megan, who did the majority of the work on this, and fairly excited to see what's coming up with our canker membrane, which we should have ready to go this summer, and then further testing of that, and take it out to the real world and start doing some more field sampling and see what we can do with it. So I'm going to stop there. So thank you very much.
We've got time for questions. Maybe
I don't remember any wood diseases 15 to 30 years ago in vineyards in BC. So where do these come from?
I don't know.
I partly answered my question when you mentioned, well, there were some, cherry orchards, apple orchards that were replaced with grapes and things like that.
Well, I think there's a couple things going on. It's really rapid growth of the industry, for one thing. So, and part of this, these diseases have been in Europe and South Africa, right. California, 20 years plus.
Could they have come in with French nursery stock? That 91 on?
Yeah, that's quite possible, because there's a few things. Just trying to remember, sodium arsenate was used as a control for these. It was banned back in, I think the last countries were Spain and France, somewhere early 2000s, that they used it up until that point. So after that, there hasn't been a really good control, chemical control for most of these. So that's one possibility. The other possibility, we're planting in apple and cherry blocks. There's another reason that we could be seeing more of these, because there's not a lot of available land left in BC unless you're in the Okanagan Valley for grape production, unless you're putting them in cherry and apple blocks. So
right. It's
a number one no-no, but people are doing it because that's what's available. So
sure.
I think steps have to be taken to know what you're planting into before you do it. The first time I saw them was in 2008 when growers called me up and told me to come down and take a look at this. So before that,
are you seeing it in central Washington, where most of it's home-rooted?
Not as much, which is interesting. They don't see the same degree of trunk disease. Now, I need to get down there and visit some more vineyards, and I haven't got down there enough yet. But reports that I have are that the problem's not as big down there.
Yeah, I don't remember seeing it there.
Yeah.
In the 80s and 90s.
Yeah. And it's still not, as far as I know, not. They do have some, not as big as what we're seeing in BC. Yeah.
Dan, I have a technical question for you. With the membrane, and with the fact that you're including some redundancy for some of the species,
right.
just in case there's some small
little
type differences there. How many species typically are you able to identify on a membrane?
If they are all present, you could identify all of them. So if one sample contained all 80 species. So
there's 80 represented on
this one right now, 80 species that are known to cause trunk diseases worldwide. Right. On the young vine decline, we have 59 species that, if they are all present, theoretically, you could pick them all up, or 1 to 59 or any
of them. So about 59 typically is what you've got. Okay.
Yeah.
And then the other, again, just a technical question: are you using the control probes to orient the membrane?
Yeah.
I know on the early ones you clip the corner.
Yes.
A couple of them, but are you now making use of those controls to help orient?
So there is software that you can create that grid, and if you have controls that you can kind of snap the grid to, and then you know that that everything else falls in line. So that's what they're there. They're also there to just show us that the hybridization worked properly in the control, because nothing shows up. It's nice to know, there's no reaction taking place for the species membranes, but the controls are all working properly.
Is there any, first of all, what are the rootstocks that are used in BC?
A lot of 3309, probably the dominant one. 10114, SO4 also out there. Those are probably the top ones I've seen. There are others, but...
Is there any variability and susceptibility to Blackfoot among the rootstocks?
Yeah. All the ones that I mentioned that are typically used kind of sit, if you had a gradient from highly susceptible to less susceptible, they kind of sit in the middle somewhere.
You didn't see a difference among those rootstocks as far as susceptibility?
We haven't done the testing for that yet. So I have one rootstock trial going on, and it looks like there is some differences showing up. I don't have all the data analyzed for that yet, but both 3309, SO4, own rooted, and 10114, and that's for cylinder carpin destructans. And yeah, there's some differences there between the rootstocks, but as I say, haven't got it all crunched yet.
Do you think climate change has anything to do with the increase? I'm segueing into Tony's talk.
Okay, yes, quite possibly. Yeah. Well, yeah, it's an interesting
talking about the change between 1990s and now.
Yeah. So I don't know what the difference in temperature, temperature would be between the 90s and now, but I know they've looked at that, and yeah, we do have an upward trend for sure. I think that would be just one of the components of the whole problem.
What's the cost for doing these?
So what for running it? It's somewhere between $7 and $10, I think. Yeah, I haven't done the numbers for a while. A few years ago I did it, but it was between $7 and $10 to run a test. The more you do, the cheaper it gets. So if you do a whole bunch, get a student, get them to do it. It gets really cheap. So.
I actually just have a question. Like what you were saying, you mentioned that some species have single nucleotide differences. Do you ever worry about unspecific binding? You said that there's no cross reaction, but what if, theoretically, could you not get something that has like one nucleotide difference binding to the wrong probe?
Yeah, that's quite possible. So when you're designing the probes, you're looking at not only the unique region, so that unique for that species, but you also have to take into account the base pair composition, GC richness, which then gives you a melting temperature. So we really try to be quite specific with the melting temperature that we select for all the probes. They have to be the same. Still, there's the possibility that you're going to have one base pair difference is a small amount of melting temperature difference. So not on this project, but on a separate project, we are able to have one base pair different probes with one base pair difference with clean separation between them. So I haven't sat down with Jose to look at all of them, all the probes, and whether we're just dealing with one base pair. Try not to do that if possible. But there are times when that's what you have and that's what you use. So, yeah.
Well, please join me in thanking Dan again for and just a reminder that Wednesday, March 6th, Dr. Tony Shaw, who's in the audience today, is going to be speaking, and the topic is Climate Change Opportunities and Risks for Ontario's Established and Emerging Wine Regions. So hope to see you all next week. Thank you.